Sexual reproduction in flowering plants. Are we not lucky enough that plants reproduce sexually? The myriads of flowers that we enjoy gazing at, the scents and the perfumes that we swoon over, the rich colors that attract us are all there as an aid to sexual reproduction. Flowers do not exist only for us to be used for our own selfishness. All flowering plants show sexual reproduction. A look at the diversity of the structures of the inflorescence, flowers, and floral parts shows us an amazing range of adaptations to ensure formation of the end products of sexual reproduction, the fruits and seeds. In this chapter, let us understand the morphology, structure, and the processes of sexual reproduction in flowering plants. Angiosperms. Flower, a fascinating organ of angiosperms. Human beings have had an intimate relationship with flowers since time immemorial. Flowers are objects of aesthetic, ornamental, social, religious, and cultural value. They have always been used as a symbol for conveying important human feelings such as love, affection, happiness, grief, mourning, etc. List at least five flowers of ornamental value that are commonly cultivated at homes and in gardens. Find out the names of five flowers that are used in social and cultural celebrations in your family. Have you ever heard of floriculture? What does it refer to? To a biologist, flowers are morphological and embryological marvels and the site of sexual reproduction. In earlier classes, you have read the various parts of a flower. Figure 1.1 will help you recall the parts of a typical flower. Can you name the two parts in a flower in which the two most important units of sexual reproduction develop? Pre-fertilization, structures and events. Much before the actual flower is seen on a plant, the decision that the plant is going to flower has taken place. Several hormonal and structural changes are initiated which lead to the differentiation and further development of the floral perimodium. Inflorescences are formed which bear the floral buds and then the flowers. In the flower, the male and female reproductive structures, the androecium and the gynoecium, differentiate and develop. You would recollect that the androecium consists of whorls of stamens representing the male reproductive organ, and the gynoecium represents the female reproductive organ. Stamens, microsporangium, and pollen grain. Figure 1.2a shows the two parts of a typical stamen. The long and slender stalk called the filament and terminal, generally bilobed structure called the anther. The proximal end of the filament is attached to the thalamus and or the petal of the flower. The number and length of stamens are variable in flowers of different species. If you were to collect a stamen, each from 10 flowers, each from different species, and arrange them on a slide, you'd be able to appreciate the large variation in size seen in nature. Careful observation of each stamen under a dissecting microscope and make neat diagrams would elucidate the range in shape and attachment of anther in different flowers. A typical angiosperm anther is bilobed with each lobe having two theca, that is, they are diphthecous. Often a longitudinal group runs lengthwise separating the theca. Let us understand the various types of tissues and their organization in the transverse section of an anther. The bilobed nature of an anther is very distinct, and the transverse section of the anther is a four-sided tetragonal structure consisting of four microsporangia located at the two corners, two in each lobe. The microsporangia develop further and become pollen sac. They extend longitudinally and all through the length of an anther and are packed with pollen grains. Structure of microsporangium. In a transverse section, a typical microsporangium appears near circular in outline. It is generally surrounded by four wall layers: the epidermis, endothelium, middle layers, and tapetum. The outer three wall layers perform the functions of protection and help in dehiscence of anther to release the pollen. The innermost wall layer is the tapetum. It nourishes the developing pollen grains. Cells of the tapetum possesses dense cytoplasm and generally have more than one nucleus. Can you think of how tapetal cells would become binucleate? When the anther is so young, a group of compactly arranged homogeneous cells called the sporogenous tissue occupies the center of each microsporangium. Microsporogenesis. As the anther develops, the cells of the sporogenous tissue undergo meiotic divisions to form microspore tetrads. What would be the ploidy of the cells of the tetrad? As each cell of this porogenous tissue is capable of giving rise to a microspore tetrad, each one is a potential pollen or microspore mother cell. The process of formation of microspores from a pollen mother cell through meiosis is called microsporogenesis. The microspores, as they are formed, are arranged in a cluster of four cells, the microspore tetrad. As the anthers mature and dehydrate, the microspores dissociate from each other and develop into pollen grains. Inside each microsporangium, several thousands of microspore or pollen grains are formed that are released. With the dehiscence of anther. Pollen grain. The pollen grains represent the male gametophytes. If you touch the open anthers of hibiscus or any other flower, you'd find deposition of yellowish powdery pollen grains on your fingers. Sprinkle these grains on a drop of water, taken on a glass slide, and observe under a microscope. You will realize you'll be really amazed at the variety of architecture, sizes, shapes, colors, designs seen on the pollen grains from different species. Pollen grains are generally spherical, measuring about 25 to 50 micrometers in diameter. It has a prominent two-layered wall. The hard outer layer called the exine is made up of sporopollen, which is one of the most resistant organic material known. It can withstand high temperatures and strong acids and alkali. No enzymes that degrade sporopollen is so far known. Pollen grain exine are prominent features called germ spores, where sporopollen is absent. Pollen grains are well preserved as fossils because of the presence of sporopollen. The exine exhibits a fascinating array of patterns and designs. Why do you think the exine should be hard? What is the function of germ pore? The inner wall of the pollen grain is called the intine. It is a thin continuous layer made up of cellulose and pectin. The cytoplasm of pollen grain is surrounded by a plasma membrane. When the pollen grain is mature, it contains two cells, the vegetative cell and the generative cell. The vegetative cell is bigger and has an abundant food reserve and a large, irregularly shaped nucleus. The generative cell is smaller and floats in the cytoplasm of the vegetative cell. It is spindle-shaped with dense cytoplasm and a nucleus. In over 60% of angiosperms, pollen grains are shed at this two-cell stage. In the remaining species, the generative cell divides methodically to give rise to two male gametes before pollen grains are shed, three-shell stage. Pollen grains of many species cause severe allergies and bronchial afflictions in some people, often leading to chronic respiratory disorders like asthma, bronchitis, etc. It may be mentioned that Parthenium or carrot grass that came into India as a contaminant with imported wheat has become ubiquitous in occurrence and causes pollen allergy. Pollen grains are rich in nutrients. It has become a fashion in recent years to use pollen tablets as food supplements in Western countries. A large number of pollen products in the form of tablets and syrups are available in the market. Pollen consumption have been claimed to increase the performance of athletes and racehorses. When once they are shed, pollen grains have to land on the stigma before they lose viability if they have to bring about fertilization. How long do you think the pollen grains retain viability? The period for which pollen grains remain viable is highly variable and to some extent depends on the prevailing temperature and humidity. In some cereals such as rice and wheat, and pollen grains lose viability within 30 minutes of their release and remain and in some members of Rosacea, Leguminosae, and Solanaceae, they maintain viability for months. You may have heard of storing semen, sperms of many animals, including humans, for artificial insemination. It is possible to store pollen grains of a large number of species for years in liquid nitrogen at a temperature of negative 196 degrees Celsius. Such stored pollen grain can be used as a pollen bank, similar to seed banks in crop breeding programs. The pistil, megasporangium, ovule, and embryo sac. The gynoecium represents the female reproductive part of the flower. The gynoecium may consist of a single pistil, monocarpellary, or may have more than one pistil, multicarpellary. When there are more than one, the pistils may be fused together in syncarpous fashion or may be free in apocarpous form. Each pistil has three parts: the stigma, style, and ovary. The stigma serves as a landing platform for pollen grains. The style is the elongated slender part beneath the stigma. The basal bulbous part of the pistil is the ovary. Inside the ovary is the ovarian cavity, locule. The placenta is located inside the ovarian cavity. Recall the definition types of placentation that you have studied in class 11. Arising from the placenta are the megasporangia, commonly called ovules. The number of ovules in an ovary may be one in wheat, paddy, mango, or many in case of papaya, watermelon, and orchids. The megasporangium ovule. Let us familiarize ourselves with the structure of a typical angiosperm ovule. The ovule is a small structure attached to the placenta by means of a stalk called the funicle. The body of the ovule fuses with funicle in the region called hilum. Thus, hilum represents the junction between ovule and funicle. Each ovule has one or two protective envelopes called integuments. Integuments encircle one or two protective envelopes called integuments encircle the nucellus except at the tip where a small opening called the micropyle is organized. Opposing the micropylar end is the salacella representing the basal part of the ovule. Megasporogenesis. The process of formation of megaspores from the megaspore mother cell is called the megasporogenesis. Ovules generally differentiate as a single megaspore mother cell in the micropylar end of the nucellus. It is a large cell containing dense cytoplasm and a prominent nucleus. The MMC undergoes mitotic division. What is the importance of the MMC undergoing meiosis? Meiosis results in the production of four megaspores, female gametophyte. In the majority of flowering plants, one of the megaspores is functional while the other three degenerate. Only the functional megaspore develops into the female gametophyte, embryo sac. This method of embryo sac formation from a single megaspore is termed as monosporic development. What will be the ploidy of the cells of the nucellus, MMC, the functional megaspore, and the female gametophyte? Let us study about the formation of the embryo sac in detail. The nucleus of the functional megaspore divides metodically to form two nuclei which move to the opposite poles, forming the two nucleate embryo sac. Two more sequential mitotic nuclear divisions result in the formation of the four nucleate and later the eight nucleate stages of the embryo sac. It is of interest to note that these mitotic divisions are strictly free nuclear, that is, nuclear divisions are not followed immediately by cell wall formation. After the eight nucleate stage, cell walls are laid down, leading to the organization of the typical female gametophyte or embryo sac. Observe the distribution of cells inside the embryo sac in figure 18 point B and C. Six of eight nuclei are surrounded by cell walls and organized into cells. The remaining two nuclei, called polar nuclei, are situated below the egg apparatus in the large central cell. There is a characteristic distribution of the cells within the embryo sac. Three cells are grouped together at the micropylar end and constitute the egg apparatus. The egg apparatus in turn consists of two synergids and one egg cell. The synergids have special cellular thickenings at the micropylar tip called the filiform apparatus, which play an important role in the guiding of the pollen tubes into the synergid. Three cells are at the chaliceal end and are called antipodals. The large central cell I mentioned earlier has two polar nuclei, thus a typical angiosperm embryo sac at maturity, though eight nucleate is seven-celled. Pollination. In the preceding sections, you have learned that the male and female gametes in the flowering plants are produced in the pollen sac and embryo sac respectively. As both types of gametes are non-motile, they have to be brought together for the fertilization to occur. How is this achieved? Pollination is the mechanism to achieve this objective. Transfer of pollen grains shed from the anther up to the stigma of a pistil is termed pollination. Flowering plants have evolved an amazing array of adaptations to achieve pollination. They make use of external agents to achieve pollination. Flowering plants make use of external agents to achieve pollination. Can you list the possible external agents? Kinds of pollination. Depending on the source of pollen grains, pollination can be divided into three types. Autogamy. In this type of pollination is achieved within the same flower. Transfer pollen grains from the anther to the stigma of the same flower. In the normal flower which opens and exposes anther and stigma, complete autogamy is rather rare. Autogamy in such flowers requires synchrony in pollen release and stigma receptivity, and also the anthers and the stigma should lie close to each other so that the self-pollination can occur. Some plants such as viola, common pansy, oxalis, and commelia produce two types of flowers. Chasmogamous flowers, which are similar to flowers of the other species with exposed anthers and stigma, and cleistogamous flowers, which do not open at all. In such flowers, the anthers and stigma lie close to each other. When anthers dehisce in the flower buds, pollen grains come in contact with the stigma to effect pollination. Thus, cleistogamous flowers are invariably autogamous as there is no chance of cross-pollination landing on the stigma. Clustergamous flowers produce a short seed set even in the absence of pollinators. Do you think that the clustergamy is advantageous or disadvantageous to the plant? Why? Getogamy, transfer of pollen grains from the anther to the stigma of another flower of the same plant, although getogamy is functionally cross-pollination involving a pollinating agent. Genetically, it is similar to autogamy since the pollen grains come from the same plant. Xenogamy, transfer of pollen grains from anther to the stigma of a different plant. In figure 1.9b, this is the only type of pollination which during pollination brings genetically different types of pollen grains to the stigma. Agents of pollination. Plants use two abiotic wind and water, and one biotic animals agent to achieve pollination. Majority of the plants use biotic agents for pollination. Only a small portion of plants use abiotic agents. Pollen grains coming in contact with the stigma is a chance factor in both wind and water pollination. To compensate for these uncertainties and associated loss of pollen grains, the flower produces enormous amount of pollen when compared to the number of ovules available for pollination. Pollination by wind is more common amongst abiotic pollinations. Wind pollination also requires that the pollen grains are light and non-sticky so that they can be transported in wind currents. They often possess well-exposed stamens so that the pollens are easily dispersed into wind currents in figure 1.10, and large, often feathery stigma to easily trap airborne pollen grains. Wind-pollinated flowers are often have a single ovule in each ovary and numerous flowers packed into an inflorescence. A familiar example is the corn cob. The tassels you see are nothing but the stigma and the style which wave in the wind to trap pollen grains. Wind pollination is quite common in grasses. Pollination by water is quite rare in flowering plants and is limited to about 30 genera, mostly monocotyledons. As against this, you recall that water is a regular mode of transport for the male gametes amongst the lower plants groups such as algae, bryophytes, and pteridophytes. It is believed, particularly for some bryophytes and pteridophytes, that their distribution is limited because of the need for water for the transport of male gametes and fertilization. Some examples of water-pollinated plants are Vallisneria and Hydrilla, which grow in fresh water, and several marine shea grasses such as Drosera. Not all aquatic plants use water for pollination. In a majority of aquatic plants, such as water hyacinth and water lily, the flowers emerge above the level of water and are pollinated by insects or wind, as in most of the land plants. In Vallisneria, the female flower reaches the surface of water by this long stalk, and the male flowers or pollen grains are released to the surface of water. They are carried passively by water currents in figure 1.11a. Some of them eventually reach the female flowers and the stigma. In another group of water-pollinated plants, such as sea grasses, female flowers remain submerged in water and the pollen grains are released inside the water. Pollen grains in many such species are long, ribble-like, and they carry passively inside the water. Some of them reach the stigma and achieve pollination. In most of the water-pollinated species, pollen grains are protected from wetting by mucilaginous coverings. Both wind and water-pollinated flowers are not very colorful and do not produce nectar. What would be the reason for this? Majority of the flowering plants use a range of animals as a pollinating agent. Bees, butterflies, flies, beetles, wasps, ants, moths, birds, sunbirds, and hummingbirds, and bats are the common pollinating agents. Among the animals, insects, particularly bees, are the dominant biotic pollinating agents. Even larger animals, such as some primates like lemurs, arboreal tree-dwelling rodents, or even reptiles like gecko lizards and garden lizards, have also been reported as pollinators in some species. Often, flowers of animal-pollinated plants are specifically adapted for a particular species of animals. Majority of insect-pollinated flowers are large, colorful, fragrant, and rich in nectar. When the flowers are small, a number of flowers are often clustered into an inflorescence to make them conspicuous. Animals attracted to flowers by color and or fragrance. The flowers pollinated by flies and beetles secrete foul odors to attract these animals. To sustain animal visits, the flowers have to provide rewards to the animals. Nectar and pollen grains are the usual floral rewards. For harvesting the rewards from the flower, the animal's visitors come in contact with the anthers and the stigma. The body of the animal gets coating of pollen grains, which are generally sticky in animal pollinated flowers. When the animal carrying pollen on its body comes in contact with the stigma, it brings about pollination. In some species, floral rewards are in providing safe places to lay eggs, as an example, in that of the tallest flowers of Amorphophallus. The flower itself is about 6 feet in height. A similar relationship exists between a species of moth and the plant Ucca, where both species, moth and plant, cannot complete their life cycles without each other. The moth deposits its eggs in the locule of the ovary, and the flower, in turn, gets pollinated by the moth. The larvae of the moth comes out of the eggs as the seeds start developing. Why don't you observe some flowers of the flowering plants or any other available to you? Cucumber, mango, peepal, coriander, papaya, onion, lobia, cotton, tobacco, rose, lemon, eucalyptus, and banana. Try to find out which animals visit them and whether they could be pollinators. You'll have to patiently observe the flowers over a few days and at different times of the day. You'd also try to see whether there are any correlation in the characteristics of a flower to the animal that visits it. Carefully observe if any of the visitors come in contact with the anthers and the stigma, as only such visitors can bring about pollination. Many insects may consume pollen or the nectar without bringing about pollinators. Such floral visitors are often referred to as pollen or nectar robbers. You may or may not be able to identify pollinators, but you will surely enjoy your efforts. Our breeding devices. Majority of the flowering plants produce hermaphrodite flowers, and pollen grains are likely to come in contact with the stigma of the same flowers. Continued self-pollination results in inbreeding depression. Flowering plants have developed many devices to discourage self-pollination and encourage cross-pollination. In some species, pollen release and stigma receptivity are not synchronized. Either the pollen is released before the stigma becomes receptive, or stigma becomes or much before the release of pollen. In some other species, the anther and the stigma place at different positions so that the pollen cannot come in contact with the stigma of the same flower. Both these devices prevent autogamy. The third device to prevent inbreeding is self-incompatibility. This is a genetic mechanism and prevents self-pollination from the same flower or other flowers of the same plant from the fertilizing the ovules by inhibiting pollen germination or pollen tube growth in the pistil. Another device to prevent self-pollination is the production of unisexual flowers. If both male and female flowers are present on the same plant, such as castor and maize, monoecious, it prevents autogamy but not ketogamy. In several species, such as papaya, male and female flowers are present on different plants. That is, each plant is either male or female, dioicy. This condition prevents both autogamy and ketogamy. Pollen crystal interaction. Pollination does not guarantee the transfer of the right type of pollen, compatible pollen of the same species as the stigma. Often, pollen of the wrong type, either from other species or from the same plant, if it is self-incompatible, also lands on the stigma. The pistil has the ability to recognize the pollen whether it is of the right type, compatible, or of the wrong type, incompatible. If it is of the right type, the pistil accepts the pollen and promotes post-pollination events that leads to fertilization. If the pollen is of the wrong type, the pistil rejects the pollen by preventing pollen germination on the stigma or the pollen tube growth in the style. The ability of the pistil to recognize the pollen followed by acceptance or rejection is the result of a continuous dialogue between pollen grain and the pistil. This dialogue is mediated by chemical components of the pollen interacting with those of the pistil. It is only in the recent years that both botanists have been able to identify some of the pollen and pistil components and the interactions leading to the recognition followed by acceptance and rejection. As mentioned earlier, following compatible pollination, the pollen grain germinates on the stigma to produce a pollen tube through one of the germ pores. The contents of the pollen grain move into the pollen tube. Pollen tube grows through the tissue of the stigma and style and reaches the ovary. You recall that in some plants, pollen grains are shed at two-celled state conditions, a vegetative cell and a generative cell. In such plants, the generative cell divides and forms the two male gametes during the growth of the pollen tube in the stigma. In plants which shed pollen in the three cells condition, pollen tubes carry the two male gametes from the beginning. Pollen tube, after reaching the ovary, enters the ovule through the micropyle and then enters one of these synergids through the filiform apparatus. Many recent studies have shown that filiform apparatus present in the micropylar part of the synergids guides the entry of the pollen tube. All the events from the pollen deposition on the stigma until pollen tubes enter the ovule are together referred to as pollen-pistil interaction. As pointed out earlier, pollen-pistil interaction is a dynamic process involving pollen recognition followed by promotion or inhibition of the pollen. The knowledge gained in this area would help the plant breeder in manipulating pollen-pistil interaction, even in the incompatible pollination to get desired hybrids. You can easily study pollen germination by testing some pollen from flowers such as pea, chickpea, Cortalaria, balsam, and vinca on a glass slide containing a drop of sugar solution about 10%. After about 15 to 30 minutes, observe the slide under the low power lens of the microscope. You are likely to see pollen tubes coming out of the pollen grains. A breeder is interested in crossing different species and often generate and combine desirable characters to produce commercially superior varieties. Artificial hybridization is one of the major approaches of crop improvement program. In such crossing experiment, it is important to make sure that only the desired pollen grains are used for pollination, and the stigma is protected from contamination from unwanted pollen. This is achieved by emasculation and bagging techniques. If the female parent bears bisexual flowers, removal of anthers from the flower bud before the anther dehisces using a pair of forceps is necessary. This step refers to as emasculation. Emasculated flowers have to be covered with a bag of suitable size, generally made up of butter paper, to prevent contamination of its stigma with unwanted pollen. This process is called bagging. When the stigma of bagged flowers attains receptivity, mature pollen grains collect them from anthers of the male parent and are dusted on the stigma, and the flowers are rebagged and the fruits allowed to develop. If the female parent produces unisexual flowers, there is no need for emasculation. The female flower buds are bagged in before the flower opens. When the stigma becomes receptive, pollination is carried out using the desired pollen and the flower rebagged. Double fertilization. After entering one of the synergids, the pollen tube releases the two male gametes into the cytoplasm of the synergid. One of the male gametes moves towards the egg cell and fuses with its nucleus, thus completing the syngamy. This results in the formation of a diploid cell, the zygote. The other male gamete moves towards the polar nuclei located in the central cell and fuses with them to produce a triploid primary endosperm nucleus, PEN, PEN. In this involves the fusion of the three haploid nuclei. It is termed as triple fusion. Since two types of fusion, syngamy and triple fusion, take place in the embryo sac, the phenomenon is termed double fertilization, an event unique to flowering plants only. The central cell after triple fusion becomes the primary endosperm nucleus, PEC, PEC, and develops into the endosperm, while the zygote develops into an embryo. Post fertilization structures and events. Following double fertilization, events of endosperm and embryo development, maturation of ovules into seeds and ovary into fruits are collectively termed as post fertilization events. Endosperm. Endosperm development precedes embryo development. Why? The primary endosperm cell divides repeatedly and forms a triploid endosperm tissue. The cells of this tissue are filled with reserve food material and are often used for nutrition for developing embryo. In the most common type of endosperm development, the PE and PEN undergo successive nuclear divisions to give rise to free nuclei. This stage of the endosperm development is called free nuclear endosperm. Subsequently, cell wall formation occurs and the endosperm becomes cellular. The number of free nuclei formed before cellularization varies greatly. The coconut water from tender coconut that you are familiar with is nothing but free nuclear endosperm made of thousands of nuclei, and the surrounding white kernel is called the cellular endosperm. Endosperm may either be completely consumed by the developing embryo, in case of pea, groundnut, beans, therefore seed maturation, or it may persist in the mature seed, example castor and coconut, and be used for during seed germination. Split open some seeds of castor, pea, beans, groundnuts, fruits, or coconuts, and look for endosperm in each case. Find out whether the endosperm is persistent in cereals, wheat, rice, and maize also. Embryo. Embryo develops at the micropilar end of the embryo sac where the zygote is situated. Most zygotes divide only after a certain amount of endosperm is formed. This is an adaptation to provide assured nutrition to the developing embryo. Though the seed differs greatly, the early stages of embryo development, embryogeny, are similar in both monocotyledons and dicotyledons. Figure 1.13 depicts the stages of embryogeny in dicotyledonous embryo. The zygote gives rise to the proembryo and subsequently to the globular, heart-shaped, and mature embryo. A typical dicotyledonous embryo, figure 1.14a, consists of an embryonal axis and two cotyledons. The portion of embryonal axis above the level of cotyledons is the apicotyl, which terminates with the plumule or stem tip. The cylindrical portion below the level of cotyledons is hypocotyl that terminates at its lower end in the radicle or root tip. The root tip is covered with a root cap. Embryos of monocotyledons possess only one cotyledon. In the grass family, the cotyledon is called scutellum. There it is situated towards one side, lateral of the embryonal axis at its lower end. The embryonal axis has a radicle and root cap enclosed in an undifferentiated sheath called coryza. The portion of the embryonal axis above the level of attachment of scutellum is the epicotyl. Epicotyl has shoot apex and a few leaf of primordial enclosed in a hollow foliar structure, the colluctile. Soak a few seeds in water, say of wheat, maize, pea, chickpeas, groundnuts, overnight. Then split the seeds and observe the various parts of the embryo and the seed. Seed. In an angiosperm, the seed is the final product of sexual production. It is often described as fertilized ovule. Seeds are formed inside fruits. A seed typically consists of seed coats, cotyledons, and an embryo axis. The cotyledons of the embryo are simple structured, generally thick and swollen due to storage of food reserves, as in legumes. Mature seeds may also be non-albuminous and or ex-albuminous. Non-albuminous seeds have no residual endosperm as it is completely consumed during the embryo development, for example in case of pea and groundnut. Albuminous seeds retain a part of endosperm as it is not completely used up during embryo development, as in case of wheat, maize, barley, or castor. New Occasionally, in some seeds such as black pepper and beet, remnants of nucellus are also present. This residual persistent nucellus is the perisperm. Integuments of ovules hardens as tough protective seed coats. In figure 1.15a, the microbial remains as a small pore in the seed coat. This facilitates entry of oxygen and water into the seed during germination. As the seed matures, its water content is reduced and seeds become relatively dry, 10 to 15% moisture by mass. The general metabolic activity of the embryo slows down. The embryo may enter a state of inactivity during called dormancy, or if favorable conditions are available, adequate moisture, oxygen, and suitable temperature, they germinate as ovules mature into seeds. The ovary develops into fruit, that is, the transformation of ovules into seeds, and ovary into fruits proceeds simultaneously. The wall of the ovary develops into the wall of fruit called pericarp. The fruits may be fleshy, as in guava, orange, mango, etc., or may be dry, as in groundnut, mustard, etc. Many fruits have evolved mechanisms for dispersal of seeds. Recall the classification of fruits and the dispersal mechanism that you have studied in earlier classes. Is there any relationship between number of ovules in an ovary and the number of seeds present in a fruit? In most plants, by the time the fruit develops from the ovary, other floral parts degenerate and fall off. However, in a few species such as apple, strawberry, cashew, etc., the thalamus also contributes to fruit formation. Such fruits are called false fruits. Most fruits, however, develop only from the ovary and are called true fruits. Although in most of these species, fruits are the results of fertilization, there are a few species in which fruits develop without fertilization. Such fruits are called parthenocarpic fruits. Banana is one such example. Parthenocarpy can be induced through the application of growth hormones, and such fruits are seedless. Seeds offer several advantages to angiosperms. Firstly, since reproductive processes such as pollination and fertilization are independent of water, seed formation is more dependable, and also seed have better adapted strategies for dispersal to new habitats and help these species to colonize in other areas. As they have sufficient food reserves, young seedlings are nourished until they are capable of photosynthesis on their own. The hard seed coat provides protection to the young embryo. Being products of sexual reproduction, they generate new genetic combinations leading to variations. Seed is the basis of all agriculture. Dehydration and dormancy of mature seeds are crucial for the storage of seeds, which can be used as food throughout the year and also to raise crops in the next year or next season. Can you imagine agriculture in the absence of seeds or in the presence of seeds which germinate straight away soon after formation and cannot be stored? How long do these seeds remain alive after they are dispersed? This period varies greatly. In a few species, the seed lose viability within a few months. Seeds of large number of species live for several years. Some seeds can remain alive for hundreds of years. There are several records of very old yet viable seeds. The oldest is that of lupine, Lupinus arcticus, excavated from Arctic tundra. The seed germinated and flowered after an estimated record of 10,000 years of dormancy. A recent record of 2,000-year-old viable seed of one date palm, Phoenix dactylifera, discovered during the archaeological excavation at Kingdom Herod's Palace near the Dead Sea. After completing a brief account of sexual reproduction of flowering plants, it would be worth attempting to comprehend the enormous reproductive capacity of some flowering plants by asking the following questions. How many eggs are present in an embryo sac? How many embryo sacs are present in an ovule? How many ovules are present in an ovary? How many ovaries are present in a typical flower? How many flowers are present on a tree? And so on. Can you think of some plants in which the fruits contain very large number of seeds? Orchid fruits are one such category, and each fruit contains thousands of tiny seeds. Similar in case of fruits of some parasitic species such as Orcobanche and Stiga. Have you ever have you seen a tiny seed of ficus? How large is the tree of ficus developed from that of tiny seed? How many billions of seeds does each ficus tree produce? Can you imagine any other example in which such tiny structure can produce such a large biomass over the years? Apomixis and polyembryony. Although seeds in general are the products of fertilization, a few flowering plants, such as some species of Asteraceae and grasses, have evolved a special mechanism to produce seeds without fertilization, called apomixis. What is fruit production without fertilization called? Thus, apomixis is a form of asexual reproduction that mimics sexual reproduction. There are several ways of development of an apomictic seed. In some species, the diploid egg cells is formed without division, reduction division, and develops into the embryo without fertilization. More often, as in many citrus and mango varieties, some of the nuclear cells surrounding the embryo sac start dividing, protrude into the embryo sac, and develop into the embryos. In such species, each ovule contains many embryos. Offering of more than one embryo in a seed is referred to as polyembryony. Take out some seeds of orange and squeeze them. Observe the many embryos of different sizes and shape from each seed. Count the number of embryos in each seed. What would be the genetic nature of apomictic embryos? Can they be called clones? Hybrid varieties of several of our food and variety crops are being extensively cultivated. Cultivation of hybrids are tremendously increased productivity. One of the problems with hybrids is that hybrid seeds have to be produced every year. If the seeds collected from hybrids are shown, the plant in the progeny will segregate and will not maintain hybrid characters. Production of hybrid seeds is costly, and hence the cost of hybrids become too expensive for the farmers. If these hybrids are made into apomix, there is no segregation of factors in the hybrid progeny. Then the farmers can keep or using hybrid seeds to raise new crops year after year, and he does not have to buy a hybrid seed every year. Because of the importance of apomixis in hybrid seed industry, active research is going on in many laboratories around the world to understand the genetics of apomixis and transfer apomictic genes into hybrid varieties.